You're welcome once again to Excellent Simplified tutorial, and today we're going to be looking at a very important concept in protein metabolism, which is the covalent backbone of protein. Now, for every building to stand, blocks are laid on each other, and that is done with the help of cement holding the blocks together before the building can stand. Protein is a macromolecule, and the building block of protein is amino acid. So amino acids are linked together for our protein to be made. Now we have over 300 amino acids, but out of the 300 amino acids, only 20 amino acids serve as building blocks of protein. Okay, now we've known that the building blocks of proteins are amino acids, but that's not all. How do we link these amino acids together, and in what sequence? Which amino acid should come before which? So the sequence of these amino acids in a protein is very, very important, because the sequence of the amino acid is what determines the protein property. Now, before we delve properly into the sequence of amino acids, let's look at the nature of amino acid. If you look at the structure on your screen, you see that the amino acid have two functional groups. On your left hand, you can see the amino group, and on your right hand, you can see the carboxylic acid group. So because of those two functional groups, the amino acids are called amino acid, because they contain amino group and a carboxylic acid group. Now, all the 20 amino acids have the same structure, but the only difference is seen in the side chain, which is represented by arrow there. That is what differentiates one amino acid from another. The side chain, the arrow group, is what differentiates one amino acid from another. And the 20 amino acids that make up protein are alpha amino acids, because the side chains are located on the alpha carbon. The alpha carbon is a carbon after the carbon bearing the functional group. So this is the nature of the amino acids. So we now know how the amino acids look like. Now, how do we link these amino acids together, and what cement or what bond are holding the amino acids in place? So that's what we are going to look at now. How we can link 2, 3, 4, 5, 6 amino acids together, and the type of bond that's holding the amino acids together. Now, the bond that we use to hold two amino acids together in this case is not cement, like in the case of block. But the cement we use, the bond we use to hold two amino acids together, is called a covalent peptide bond. A covalent peptide bond. And how is this covalent peptide bond formed? How do we join these two amino acids together using a covalent peptide bond? Now, from the image on your screen, you can see the first amino acid and the second amino acid. Now, if you can see, on your right hand side of the first amino acid, you're having a carboxylic acid group, C double bond to oxygen and OH. Then on your left hand side, the left terminal, the end terminal of your amino acid, you're having N and 2 hydrogen, nitrogen and 2 hydrogen. Now, the formation of peptide bond coming together of these two amino acids is a condensation reaction, meaning water is removed. So OH is taken from the first amino acid, and H is taken from the second amino acid to form your H2O, water going out. Then you can join the remaining bonds. We'll take practical examples. And if you don't get this, please pause the video and listen again. Now let's look at how we can form different oligopeptides. Now, if you join two amino acids together with a covalent peptide bond, what you are having is a dipeptide. If you join 3 amino acids together, what you are having is a tripeptide. If you join 4 amino acids together, what you are having is a tetrapeptide. If you join 5, pentapeptide. If you join 6, hexapeptide. If you join 7, heptapeptide. if you join 8 octapeptide, if you join 9 nonapeptide, if you join 10 decapeptide, and so on. Now we've known that a dipeptide contains 2 amino acids, and we know also that if you are joining 2 amino acids together, you remove OH from the carboxy end of one and hydrogen from the amino end of one to form your peptide bond in a condensation reaction. Water is given off in the process. Now, for a dipeptide, you're joining 2 amino acids together as shown on the screen. And for a tripeptide, we are going to, it's still the same process. Remove your hydroxy group from the carboxy end of the last amino acid and hydrogen from the amino end of the next amino acid. You join them together, form your peptide bond. So for tetrapeptide, you do same. So the process remains the same. Whether it is a dipeptide, it is a tripeptide, it is a tripeptide, the process remains the same. Just understand that you are removing OH from the carboxy end of the last amino acid and you are removing hydrogen from the amino end of the incoming amino acid. Do you understand? If you don't understand, please pause the video and look at this structure on your screen, and I hope you get. Now let's take for example the peptide bond from the dipeptide glycine and alanine. As you can see on your screen, glycine on the left hand side, alanine on the right hand side. So what was just done there is OH was removed from the carboxy end of glycine and 1 hydrogen was removed from the amino end of alanine, and they were both joined together to form your dipeptide. So this is a dipeptide between glycine and alanine. For other amino acids, the process remains the same. Just know the structure of your amino acids and you can do that. Now take some time to look at this structure on your screen. Is this a dipeptide, a tripeptide, a tetrapeptide, or what? You can pause the video and you analyze this. So I'll give you some time to analyze this. After that, you can come back and you can check if you were right or you were wrong. But just as you're doing that, try to identify the position of the peptide bond. So if you can identify the positions of the peptide bond, you will actually know which peptide it is, either it is a dipeptide, tripeptide, or tetrapeptide. Okay, so you're welcome back. So what was your answer? If your answer was a dipeptide, you are actually wrong. This is a tripeptide because it contains 3 amino acids. 3 amino acids. So the 3 amino acids, tripeptides have 2 peptide bonds linking the 3 amino acid residues together. So that can be seen in the diagram as shown. Please, if you have any question, you can drop it in the comment section. In our next video, we are going to be looking at how we can sequence amino acids. Please, as you watch, like and subscribe so that you don't miss out on any of our videos. Thanks for watching. See you in our next video.